that you can do and have a look at. And then the 4th of August we can quickly discuss if you have any questions regarding the exercise, the exam, and if you have any questions regarding the questions we are going to ask within the exam. I updated this slide on the in Ilias as well, so if you want to have a look at it, just go online and all the dates are correct. Okay, so let's start with the unit operations lecture. So for the learning goals for this lecture, we have defined 3 important points. So first of all, you should be able after this lecture to explain what a process chain is for simple workflows. You will be able to classify unit operations for different workflows, so what kind of unit operations there are, and also how you can classify them in themselves. And last, you have to be able to identify relevant unit operations for microfluidic workflows. Yeah. So let's start with the terms process chains and unit operations. So you probably have never heard of it, and that's why we define it on this first slide. So a process chain is a sequence of required unit operations for implementation of a workflow. So if I want to break this down, it's always helpful to have a look at a graphical display of a process chain. So in principle, you have something that you put into your system and something that you get out of your system. And the process chain covers all the steps that are necessary to come from your input to your output of your system. And it's displayed here, and I will do together with you 2 examples on the next 2 slides that you get an impression on what it really means. And a unit operation is one single step of the complete process chain. So if we look at the example on the right hand side, we give something in, let's say some liquids, some reagents, we transport the liquids, we do some kind of metering where we determine an exact volume, and then we do some mixing of the volumes and we get a mixture like a PCR reaction or another kind of reaction out of the system. In this lecture, we only cover 4 types of unit operations, but there are heaps more. I guess those 4 are the most intuitive ones because you always need to transport liquids in a certain way, for example. And they are also, the most prominent one that are the most often used and required. So let's start with a very simple example. An inkjet printer. And this printer is depicted on the right hand side. So the basic principle of it is that if you want to print something, you add a droplet of a certain ink or color onto your paper, and by doing this repetitively, you will get some kind of image, like here depicted a horse. And so what you have to achieve, so what your process chain has to achieve is that it has to dispense micro droplets of a diameter of around 50 micrometers. And as you can see here on this side, I've left one unit operation open, and maybe after we having discussed the principle, the working principle, you will be able to add the missing unit operation. So let's have a look on the small sketch on the right hand side. So what you can see here are 3 different steps that happen. You have some kind of heating element, you have a tube that contains the ink, and you have a nozzle at the bottom where the liquid gets squeezed out. And what happens in the second step is that the heating element gets heated up, and you see that the air bubble, which is positioned here, increases because of the ideal gas law. So if you heat up, it will expand. And this finally leads, if you continue doing this, the bubble will further increase and grow and will then displace the liquid out of your tube and via your nozzle. And this is how this droplet gets generated. So what we do first is there's liquid inside your, so that's your in status, there's liquid inside your tube. And by building up this different, or by building up this air bubble, you get a defined volume which is underneath your air bubble, right? And that's the valving procedure. So the liquid of the top is separated from the liquid on the bottom. At the same time, this second unit operation here happens. Do you have any idea what it could be? So one of those four. Metering, right. So by doing this valving process, we have a defined volume which is separated from the upper volume, and that's the metering principle. And last but not least, by further increasing the air bubble, we push out the liquid, and that's our way of transport in this example. Okay, so the second example is a cartridge developed by us in our institute, and it's a very simple one. So we're doing a PCR on this cartridge, and it's depicted on the right-hand side. So you can see that there are microfluidic structures on it. There's a channel moving from the upper chamber towards the outer chambers, and there are several reaction cavities. So the advantage of having different reaction cavities is that in every or each of them you can add different reagents like primers or a different master mix lyophilized form. And by this you can use every single cavity for doing another type of assay. So for example, in the first cavity you could measure if the patient has SARS-CoV-2. In the second cavity you could measure if the patient has influenza. And in the third cavity you could check for something else. So the workflow, once again, is depicted on the right-hand side. So the first thing that you have to do is you have to add some kind of sample from the patient. You then start rotating the cartridge, and you make use of centrifugal forces and distribute the liquid along this structure here. And by having this waste structure here at the bottom and defined volumes for each of these cavities, you will end up with the same liquid volume in each of the reaction chambers, which is important for a comparison between the chambers. And then in the last step, you accelerate, and the liquid gets transported in the final reaction chambers, where you can then do the readout by doing a PCR, for example. So if we look at this process once again, what are the steps that we are doing? Do you have any idea? Sorry? Transfer liquid, right? What else? Sorry?
Valving.
Yeah. And last? So this is so first, obviously, the transport of liquids. We then have this metering structure that I added here. So I talked about it, that you have the same volume in each of the cavities, and you could also use what you said, valving, because the liquid stops at this position. So this would also be a valid option, or you can even say both at the same time. And then I also added here mixing, because if you move now the liquid into the last reaction cavity, you have to mix the sample with your reagents. And that's why you need to implement some kind of mixing procedure. Okay, so that's the principle of process chains. As you have realized, it's not always, when we were discussing, it's not always that fixed. It really depends on how you have or how you analyze your system. But in most of the cases, and especially in the exam, we will have very defined examples where it's clear which unit operations are required and in which order you have to put them. Okay, so then now let's really have a closer look at the 4 different unit operations that we want to cover within this lecture. So we will start with the most important one, which is transport of liquids. And before I go into detail, I quickly want to explain what you will see in the next slides. So on the right-hand side, this is what we did during the semester. And our idea was that we are highlighting the chapters that are important for the concept that I present. So you will see later on, if we are talking about capillary forces, obviously the surface effects lecture is of importance, and this should help you to get an idea of why you needed what we did during the complete semester. So yeah, let's start with transports of liquids. So do you have any idea how can liquid be transported? What are the forces that could be relevant to transport liquids? I think we talked a lot about them during the semester, and I think that you have some ideas about it. Then what force?
Gravitational force.
Gravitation force, yeah, right. Or like gravity field, like centrifugal, yeah.
Pressure forces.
Sorry?
Pressure forces.
Pressure forces, how could you build up pressure?
Gravitation, so as I take like the chip that I have, I can.
Right, that's the centrifugal pressure. What else could there be? If you think very pragmatic, very practical, how do you build up pressure in the macro world?
Compress things.
Right, you have some kind of pump that pushes your system. Okay, what other way of generating pressure did we have a look at? Sorry?
Using
Heat, yeah. Kinetic pressure, yeah. I would rather put that in the direction of the pump system. But if we look at the surface effect lecture, for example,
Surface tension.
the capillary forces, yeah. So we use the contact angle and the surface tension, right? Okay, so there are more concepts available. Today we will just talk about these 5. So there's a capillary-driven system of moving liquids. You have this displacement system, which is rather in the direction that you were talking about with pumps. We have the centrifugal-driven approach, which is obviously the most important for us here at the institute or in the lab of Professor Zengerle. You can also make use of electrokinetics. That's what we also discussed in this electrokinetic effects lecture. There are different ways of using this effect. And there you could also use acoustic effects. I really don't want to deepen the discussion on this topic because it's rather a side aspect of microfluidics. So yeah, let's start with capillary-driven microfluidics. So obviously, all you need basically to understand the propulsion force here is the surface effects lecture. Because the liquid is purely transported by capillary pressure. And the most prominent example where this kind of technology is used are lateral flow stripes. And they make use of it by using porous membranes. So within these membranes you have very tiny holes, and as you remember, the smaller you get, the bigger the capillary force gets, because we had this term 1 divided by the dimension, which is proportional to the capillary force. And this is why those small holes, they lead in quite big capillary forces, and that are often, not always, but often used for capillary-driven microfluidics. So if you think about this concept, what could be a very obvious reason why would we use such kind of systems.
Maybe because you don't need any extra material.
Right, yeah, so you don't really need any external actuation. You just add a liquid, it moves through your structure, and then you get some kind of result. And this is, if you think about SARS-CoV-2, I guess all of you have done the testing where you put some liquid yourself onto some kind of lateral flow test and then got your result if you're positive or negative. So this is really one of the crucial advantages of working with capillary-driven microfluidics. It's simple and basically everybody can do it at home. What could be the disadvantage on the other side?
Maybe we don't have much control over the flow.
So the controlling is much harder, and obviously also the opposite. If you have very complex workflows, this is almost impossible to achieve with capillary-driven microfluidics. Okay. And so obviously the most obvious example here is antigen testing or even pregnancy tests. I mean, those are the two famous examples where microfluidic driven by capillary forces is often used. Okay, then let's move on to the displacement concept, which is probably the most obvious when looking or when coming from the macroscopic world, where you usually always have pumps that drive your liquid through some kind of networks or some turbines. And in this part I want to quickly discuss three different concepts with you. The first one is a peristaltic micromembrane pump, and it is depicted here in the center. So what you can see is that there are three controllable elements that are these dark colored rectangles here at the top, and by activating them you can deform your membrane, so like you just showed me a second ago. And the idea here is that the liquid can move through this white colored areas basically. Opening or close, you can prevent or allow the liquid to move through your structures. The way how you control these controllers is not really of importance. You could make use of pneumatic controlling, but you could also use some kind of piezoelectric or thermal or electric control. So what happens in the first step is that the liquid, so the first membrane is deflected and the liquid can move into your structure until this point. If you then open the second membrane, it can move further and enter and get sucked and pumped until this point here. And after that, you close the last one of the first one of the controlling elements, and that's why the liquid can't move in the backwards direction anymore. So it's trapped between the left and the right controller. You then open the right controller, and I guess now you really see that the liquid, by pushing down the middle controller, you can push the liquid through your system out to the right direction. So if you want to simplify this concept, you can think about a tube, and if you now push, and the tube is fully filled with liquid, if you now squeeze it in one position and move along the tube, you will just push the liquid out of your tube. And that's the same concept here, except that it's not continuous, but that you have three defined volumes or valves that you use to push the liquid out of your system. So obviously, if you have three controlling elements, the more controlling elements you have, the harder it is to work with it. And that's why people invented other kinds of pumps. So here you can see that in this sketch here that you only have one controlling element and two valves that are to the left and to the right of it. And that's what's typically called a displacement pump with check valves. And the idea here is that you move this, or that you deflect your membrane up and down in this position, and that the valves only allow to push liquid in one direction. So if you move in upwards direction, liquid gets sucked into your structure from the left-hand side, and the valve 1 allows flow in this direction. If you now press it down, the valve, as you can see how it is designed here, will keep closed, and there's no liquid movement in this direction, but there is liquid movement via the second valve because it allows flow in this direction. And by now, quickly moving up and down with this membrane, you can generate an almost steady or continuous flow in your microfluidic system. And this can be further enhanced by replacing the valves by microfluidic elements. In this case, it's just a diffuser to the left and to the right, and the diffuser has a preferential flow direction. And the principle is the same. You have one controlling element that can be deflected, and that increases or decreases your volume in the cavity. But thanks to this preferential flow direction, there is a bigger inflow from the inlet side and a bigger outflow to the outlet side. And yeah, this element only makes use of this diffuser effect so that you have an increase or decrease of your square area, so that the area at this position suddenly gets bigger. And in this case, obviously you always have a flow into the control chamber from both sides and an outflow into both directions. But it's just, the importance is that you know that the inflow is stronger from one side and the outflow is stronger into another side. And that by then repetitively moving the membrane up and down, you will also be able to transport liquid in one distinguished direction. Okay, so just once again, the idea is to reduce the number of control elements and to reduce the number of elements at all, because the simpler your system, probably the more reliable it works and the easier it is to design. So one example where some kind of a pump is used is here presented by this picture. It's from Cytina, and what they did is that they have some kind of flexible membrane, and it's similar to the inkjet principle that we just discussed, except they don't use an air bubble. An air bubble, they use the flexible membrane, they squeeze it, and by that they can dispense a defined volume of liquid, and they use this concept to print single cells. So they check if there is a cell in this droplet, and if there's not a cell, they remove it from the system. And by this principle, you can really have do analysis of single cells and understand what happens in a single cell and what differs different cells, for example, in tumor analytics. Okay, then let's move on to the centrifugal concept that you mentioned earlier. So obviously here the driving force is the centrifugal force. There are other forces involved as well that are typical for centrifugal systems. We will talk about this in detail in the later lecture. Let's quickly have a brainstorming about the advantages and disadvantages of this system. What could be a positive point compared to the other solutions? So think about it as the chip is always rotating. What do you think is the connection to the outer world?
Just one control element.
Sorry?
I have one control element.
Yeah, right. So this is that's it. So you only use centrifugal force basically to do some kind of liquid movement, and the idea is that you enter your liquid and then you press start, and that means that you don't require any tubes to connect your disk to the outer system. So if you're working with a pump, you have to connect it somehow with your chip and pay attention that everything is tight and that there's no leakage. And for the centrifugal approach, you don't really need that. A second positive point is that you can reduce the issues with bubbles because you always have a gravity field. And that means liquid gets separated from air simply by the gravity field. So if you have a lower density, the force is lower, and that's why the airs will move radially inward and the liquid moves radially outward. Yeah. Typical limitation is the geometry, so as you can imagine, there is a limited size that you can work with. So you can't really use very huge chips, you are limited by the device that you're working with. So an example is one of our spin-offs, SpinDiag. So they use this kind of technology to do fast COVID detection at the point of care. So they are working with this system in a clinical setting where doctors have to get quickly information if a patient is positive or negative. So then the next concept is for transport of liquids electrokinetics. And here we use the electrowetting effect to transport liquids. As a reminder, we discussed this very briefly in the surface effects lecture. Basic idea is that you have different electrodes underneath the liquid, and by applying a voltage, you change the contact angle on one side of your droplet. And by this, you generate an imbalance of pressures, and you can move the droplet from one electrode to another. Yeah, so the big advantage of this solution is that you are able to handle very small volumes, and that you can have basically a generic setup of electrodes and do whatever workflow you want to do. And that's meant with this high flexibility. The disadvantage is that it's hard to manufacture the chips, so you require specific coatings and coating technologies. And the influence of the liquid properties is not irrelevant. So depending on your contact angle, your system will behave differently. So there are already products out there that use this technology. I want to give you a quick impression on this system by discussing the video. So what you can see here on the right hand side is an exemplary chip from GMark, and every square field is basically a single electrode. And what you can now see is that they use this technology to move different liquids along the electrodes, and by moving, for example, they achieve better mixing reliability. And then they are able to distribute it on smaller electrodes, make smaller droplets. Most of the time, if they move the liquids back and forth, that's their approach to mix liquids. yeah, in the end, I guess they will move all the liquids back to this detection fields, and they do some kind of detection, electrochemical detection. So keep in mind, all the movement, that's only due to change of contact angle, just by applying electricity. So then we have electrokinetics. We had the lecture. There are two different forces that are relevant: electrophoresis, electroosmosis. You probably remember it. Here we talk about the electrical double layer that leads to the electroosmotic flow, and you also have the attraction force from the electrophoresis. If you think back to last week, what could be a big advantage of, in particular, electroosmosis? Think about a flow profile.
You can transform like in a rectangular sort of.
In a straight line, basically. Yeah. That's so you don't have this influence of the Taylor dispersion, which Nicholas presented, I guess. And so you can move all analytes at the same speed through your channel. The disadvantage is the heat generation. So always, if you're working with electrical systems, there's the risk that you generate a lot of heat, especially for this small small volumes we are talking about. And another thing that could happen is that there's a change in pH value due to electrolysis at the electrodes. So this is one of the first microfluidic chips that really was commercially successful. It's from Agilent. It's quite a simple system or chip that you can see on the right hand side. So you have some wells for the samples, which are the blue ones and the green one, and you have some other wells for a gel. And what you do in this chip is a gel electrophoresis. So you separate, or gel electrophoresis is used to separate DNA fragments via their length. So the longer a DNA fragment, the longer it takes to migrate through this chip. And by adding fluorescence, you are able to detect the time when DNA fragments are moving through the channel. And to do so, you have to pay attention to the effects that we discussed last week. So you have to pay attention to the electrophoresis, but also the electroosmosis, or the flow from electroosmosis. Okay, so to sum it up, we had those four transport principles. I guess what's really important to understand those is the fluid dynamic lecture, but also surface effects and electrokinetic effects. But if you have really understood those lectures, the principle on how the transport works should be quite clear to you. Okay, then let's move on with valving. So valving is basically how you can stop or prevent a liquid flow or allow a liquid flow. So any ideas? What could be a valving concept? Let's start at home. So if you want to have water flowing into your sink, what do? You just open some kind of lever, and I guess there you usually have some geometric element that blocks the water from flowing through your system. And in general, that is an active way of controlling liquid flow. So you have an external actuator, but you can also control it passively. And this active-passive classification will come up for every type of unit operation that we're talking about. So the passive one always has the advantage that you don't need any external actuation. In general, the active valuing concepts are more reliable and easier to implement. So the first one, once again, you can make use of capillary forces to stop liquid movement. It's depicted here. So if you have a very small channel and it suddenly gets bigger, you have a pinning effect at this position because of the capillary forces, which prevents the liquid to move further on. I guess it's easier to imagine for the hydrophobic valve. So if you have a hydrophobic patch here, for example, then you would generate a force. When the liquid moves onto this, you would have a force that pushes the liquid back into the initial direction because of the contact angles at the walls here. So quite simple. If we look on it in detail, remember what I told you in the capillary lecture. The contact angle hydrophobic would look like this, and you would then have a force directed in these directions. And that basically means you need a higher force to overcome this force to push the liquid through your system. And once again, this concept is always when you make use of capillary forces, it's the easiest and most of the time cheapest approach to do what you want to do. The disadvantage is the reliability. So as you can imagine, it's really important how big your channel is. So if it gets bigger, your counter pressure will decrease. If it gets smaller, your pressure will increase. And this combined with the buffer you're working with and the influence of the buffer makes your capillary valve not very reliable. There's another concept called a Tesla valve, which is also a passive valve, so there's no external actuation. And it is used for continuous approaches. And it could also be part of some kind of pumping principle. The idea is that you have a preferential, or that you can have a preferential direction with the Tesla valve. Why is that the case? So if you pump gas into your Tesla valve or liquid from the left-hand side, it's the gas or liquid doesn't really like to move in perpendicular direction via this channel. If you look on it from the other side, it's way easier and way more evident for the liquid to move in both channels. And this basically leads to counter pressure at this position here. So if you exaggerate the design of the capillary valve, so this is your channel, this is the other channel. So if you really change the angle from 90 degrees towards a bigger angle in this direction, you will have a flow which moves against the flow direction of the other channel. And this will reduce the total flow of your system. Yeah, is that clear, the idea? So the liquid will separate at this position, it will move alone, and then you will push it out of your channel in this direction. And this will definitely work against the flow that comes from this direction and basically reduce the total flow out of your system. So this means that in this direction the flow is faster because it will simply neglect this side arm, and it's slower for this reverse operation direction. Okay, then for the active valves, it's way more simple what you are able to do. So what we see here in the picture is a deformable microfluidic chip. The blue one, you have a glass slide to cover the chip, and you have some kind of actuation structure on top of your chip. And what you can do now is by making use of this actuation chamber, you can deform the chip and fully press the blue material onto your glass slide. And then you have a closed contact, and obviously liquid can't move through your channel anymore. And that's why they work with this circular shaped form. So if you look at the bottom, it's the inverse. So you have a very thin film for the actuation element, and by now, for example, heating up this actuation channel, you can push this thin film right along your microfluidic structure and thereby close the system. So compared to the other two approaches, this is very reliable. So if the channel is closed, it is closed, and it's impossible for liquid to move through your channel. However, if you
Can you explain that before?
Yeah.
I didn't understand.
Yeah, sure. Control it. So let's say this is the first. This is second. And what you basically do is by applying pressure here, you deform the foil. And it will take the shape of your channel. Right? And the liquid moves through the plane. And if you now close it by pushing this membrane or this film into your channel, the liquid can't move through it anymore. So if you look from the top side, it would rather look like this. So you have a microfluidic channel, and you have some kind of actuation channel, and you now push down the actuation channel in this area, and then the liquid can only move up to this position or to this position because the channel is not blocked. Okay?
Can I ask a question?
Yeah, sure.
How does this work in the first, like in the pi day? Because in pi day the actuation channel is above the fluid channel.
Yeah, so that's the first one. The idea is that you can deform your fluid channel.
Oh, okay.
So you just squeeze together your the blue colored element, and it then just gets pressed onto the glass slide.
So in the above figure you are controlling the fluidic channel, and in the bottom one it's the actuation channel.
Right. So in the in picture A, you work with the blue one, which is the formal. B, it's the red one.
But which one is better?
Sorry?
Which one is better over the like?
There's no preferential solution. It depends on what you want. And on your materials, obviously. If you have, solid fluidic structures, you can't deform them. You have to deform the sealing foil. And if you have a deformable chip, why would you, with a glass slide, why would you, you can't really push the glass inside. You have to deform the chip. Okay, that's it for welding. Let's move on with mixing. So any ideas how could we do mixing? it. Think about how do you mix your coffee, or if you mix some drinks, how do you do it?
Like standard force fermentation.
Yeah, so you're just doing some stirring, or you also can mix them by just shaking them. And there's one lecture at the start, diffusion and heat transfer, which is the fundamental principle of mixing. So you just wait and let diffusion do the work. Because we're on a micro scale, this sometimes is enough, but often you work with distinguished mixing solutions.
But sometimes diffusion is really slow.
Yeah, diffusion is often too slow. Yeah, that's definitely the case. Often you use some kind of mixing principle. So you see, once again, we have passive. There's a question online, so if turbulent flow is a viable option. So in principle, you can work with turbulent flow, but if you remember the fluid dynamics lecture, it's not that easy to achieve turbulent flow within a microfluidic system. Okay. So once again, you have passive and active mixing procedures, but there's also batch and continuous mixing. Batch mixing is you have two volumes, for example, of a defined amount. You mix them together. Continuous mixing is that you mix, that you have a continuous flow which is mixed over time. Yeah, so I was already mentioning the mixing by diffusion. That's what you can see here on the top. So it's a mixture of two liquids, one with fluorescent and one without fluorescent particles. And you can see that after a certain time, so in this case after 10 seconds, the liquids are mixed together. But keep in mind, it's a very slow procedure. It depends on the liquids that you're working with. And usually we don't do mixing by diffusion only because it takes too long. So that brings us to the next approach. So you can make use of surface acoustic waves. That's similar from the idea to an earthquake. So you just make the solid phase shake, and by that you can mix reagents. It's a similar picture. You have a drop on a surface with two reagents. If you now apply this surface acoustic waves, you are able to mix your liquids quite fast and efficiently. So, and then there is some kind of mixing by stirring that you mentioned at the start. So in this case, there are magnetic particles within your liquid, and by making use of a magnet, you move the particles up and down, and that's how you achieve some kind of stirring protocol that helps to mix your reagents from when they were unmixed until they are mixed.
Are they magnetic particles or fluid particles that have magnetic particles?
So they are usually, that depends on the particles, but in this case I think they were just magnetic particles. And I don't even know if we have ever applied if we have ever had fluidic particles only. So usually we use the magnetic particles for some kind of biological assay or reaction.
But are they solid particles or how do you?
Yeah, they are solid. So you just have a microsphere, let's say of 1 micrometer, and you mix it by just moving these particles through your liquid. Yeah, obviously this can lead to damage regarding the cartridge. So if you scrub along on plastic with metallic components, this can lead to failure or destruction of the cartridge. So that's one thing I want to discuss a little more. So this is passive continuous mixing, and it's called multilamination. And here the idea is that what is depicted here down in the picture, on the down side, and you have two reagents, and the mixing time only depends on the area of contact between the two liquids. So if you increase the contact area, you will also decrease the mixing time. And that's what you can see in this picture. They are increasing the LAMI, or they are building up multi-lamination systems where you increase every time the surface of contact between the two faces, and the bigger or the faster or the more laminates you build up, the faster your liquid gets mixed. And obviously this is that's a very simple concept to work with, but to implement it in real life is rather complicated because you have to have many inlets for each of these lines that you see here. Okay, so that was rather quick regarding the mixing, but I think mixing is quite intuitive because you know it from the real world. And I mean, this is something I can only recommend you if you try to understand the microfluidic concepts. Always go back to the macroscopic world. Many of these microfluidic concepts are simply transferred from the macroscopic world to the microscopic world, like stirring or like shaking liquids and so on. So there are many similarities. Okay, and so the last chapter is about metering, and I mean, this is something that I can never stress enough. For each of the unit operations you have passive and active concepts, and the idea is always the same. Passive concepts don't need any external actuation, active concepts need it, and if you have understand the difference between passive and active, you are also able to directly find benefits and disadvantages of your concepts. Here's this differentiation between geometric and interfacial metering. Any idea what interfacial metering could be? Okay, then we'll get to it in a second. Geometric metering obviously is rather clear, right? So metering, you want to have a defined volume. You make use of geometry to have this defined volume. So you just construct a design that can take up 1 microliter. If it's filled, you have your microliter that you wanted to measure. So that's what we had at the start, the example of the centrifugal chip. So the idea here is that you push liquid into your system, you remove the supernatant, so the liquid that you don't require, and you have a defined cavity here which can contain a certain amount of liquid. And this liquid is held in position because you have pressure that builds up because of the compressing of the gas, and you also have a capillary restriction at the bottom, and these two forces and pressures will be in balance with the centrifugal pressure at this position, and that's how you meter your liquid in this case. So what I said before is valid for this concept as well. It's a passive concept, so it's easy and cheap. Nevertheless, the precision is quite good in this system, but it only works with centrifugal fluids. The second concept that is about active geometric metering is presented here. So the idea here is that you have controlling channels which are highlighted orange, and you have channels where the liquid moves, similar to the idea that we presented here. And by closing the valves, you can, or by closing the valves and opening them, you can move liquids. And as you can see, there are cavities that have a defined size. And if you now fully fill these cavities and close the valves, you know exactly which volume is here in between. So it's just an extension of this principle. If you add another control element, and you close both valves, you know exactly how much liquid is in between those two, if you know how you designed the chip and what geometry the chip has. Is it clear? Okay. So that's, I mean, this is very intuitive. It's basically similar to when you're cooking and you're adding liquid to a certain geometric cup, or yeah, you know exactly what volume it has. So, and the last concept is this interfacial metering concept, which is quite popular in the microfluidic world because it's something that you can't really do in a macroscopic way, and it is one of the big advantages of microfluidics. And the idea here is that you have two phases, some kind of oil and some kind of other liquid like water, and you have or you generate emulsions similar to when you're looking at a salad sauce and you see those small bubbles from the oil and from the water. That's basically the same concept, except that based on microfluidic, the knowledge you have about the fundamentals of microfluidics, you can design your system so that you exactly know how much liquid will be in each of the droplets. And that's meant by this interfacial, or by the wording interfacial. So you don't have defined geometric volumes, but you have defined interfacial volumes, because you know exactly how to generate the droplets, and thereby also knowing how big these droplets get. I think Niklas will talk a little more about it in the next lecture, because it's quite an interesting concept. Yeah, that's just for you as information. There are different ways to generate droplets. Some of them are presented here. So as you can see, sometimes the liquid comes from the side, sometimes it is at the center, and it's just sheared by an outer flow. I think Niklas will talk about it next week in more detail. Yeah, do you have an idea what could be an advantage of this concept?
Precision.
Precision, yeah. What else?
It's not complex.
Mm-hmm. It can be complex, it can become complex, but in general, if you have a running system, it works quite fine.
That's fast.
Mm-hmm. And maybe if you think more in the direction of, or let's put it the other way around. So what do you think? Is it easy to make many droplets, or is it rather hard? It's easy, right? And if you think about the manufacturing of geometric structures, you have to manufacture each well individually, at least one time for your master tool, and then you can use mass production technologies, but you have to manufacture it once. Here you only have to manufacture this structure here, and you can generate hundreds of thousands of droplets and have a way better resolution compared to the geometric approach. Yeah. So this negative point, this is maybe more a reminder to the surface effects lecture. If you remember it, the stability of the droplets, or the bubble that we discussed, the soap bubble, depends on also depends on the surfactants. And this is the hard part about this droplets microfluidics. You have to find good combinations of oil and water and surfactant, which is added to the oil, to get stable droplets and to keep them stable. Okay, so I guess that's it for today. Let me quickly, sum up what we did. So if you come back to the learning goals, it's really important that you're able to explain what a process chain is. And also if we give you an example, that you are able to determine what kind of unit operations are required to fulfill the function of the system and to get an idea of the complete process chain. You should be able to classify unit operations for different workflows. And I think the most important part, and which makes it really easy to you for the future, is that you understand the difference between active and passive, and also for the different types that we discussed today, that you have an idea of what is batch mixing, what is continuous mixing, what is interfacial, what is geometric. But if you have understood the active-passive difference, you have understood, 80% of the concepts. And the last thing is that you are able to identify relevant unit operations. So that's once again the idea. We give you an example or a workflow that you have to automate, and you have to think about it and tell us what would be a unit operation that is required to fulfill this workflow. Yeah. I guess that in the exercise this will be discussed a little further. And yeah, so I think that's it for today. Any questions? Any comments? Okay, then thank you very much for your attention. I think John is taking over today.
Yes,
We had exercise last week. And yeah.
should we think we have like a short break, like 5 minutes? Are you guys okay to continue, or should we have 5 minutes break? Okay. So we can digest a lot of information.